Metabolon is the global leader of metabolomics research. We collaborate and partner with academic institutions, government institutions, as well as pharmaceutical industries to really drive this understanding of metabolomics. Metabolomics is the study of small molecules in a biological system, and this could be a traditional biological system like you think of cells or tissues or plasma or urine, but could also be something like the food that we eat or even the medicines we take. These are all collections of small molecules. Metabolomics is the science of studying those small molecules. We hear over and over again that metabolites are incredible, so useful, but how do I drive insight from this? We often talk about scientific research as a journey, right? There is any phase of a journey where you're going to have a discovery phase, where you're trying to figure out what the biomarker is, you're trying to figure out what the disease signature is. There is this natural progression over this scientific arc to go from certain types of mass spectrometers, untargeted / discovery methodology, through to more targeted approaches. Historically and classically, these have used very different types of mass spectrometer methodologies, which have unfortunately not been very translatable in terms of the data itself. And so ultimately what has happened is that as you're making this transition into starting validating and creating these targeted methods, you end up having to start from scratch almost, because you can't use the data necessarily from your discovery mass spectrometers.
So we were interested in the Stellar Mass Spectrometer because it had the ability to do HCD and CID fragmentation. So we have a huge library that we've built on Thermo Scientific Orbitrap-based instruments. In the path from discovery to potentially clinical, you often move through using a triple quad because of the specificity and the sensitivity of those things. But that requires a real rebuild of a lot of your analytical information because it's a different type of fragmentation. So we were interested in seeing whether the functionalities of the Stellar would actually allow us to take a lot of our analytical learnings from what we've done on those Orbitrap instruments and move it seamlessly into a more targeted analysis. The learning curve was actually really fast for somebody who is very used to sort of the thermo environment. The software is very similar. A lot of the same concepts transfer over, whether it's the concept of AGC or what happens in an HCD cell. So the learning curve, if anything, was very, very short.
We want to stay at the leading edge of technology, and so we are constantly on the lookout for new technologies that will enable us to drive into producing a better, more streamlined product for our clients. So we were in discussions with Thermo Fisher Scientific, and they brought our attention to the Thermo Scientific Stellar Mass Spectrometer, and that this could potentially be an instrument that could help bridge that gap between the untargeted discovery phase into that targeted. So this was our interest in looking at this instrument, was does this help bridge that gap? Does this make the process more efficient?
So before we started, we had heard from the team about the scan speed, but until I actually started operating the instrument itself, I didn't really understand where those benefits came in. So because of that scan speed, we were actually able to do full scan positive, full scan negative, fragmentation scans in positive and negative with multiple target ions throughout our entire chromatographic run. And until we did that, I didn't really understand how powerful that type of analysis can be. From one working day, from the first thing in the morning to the time we left in the afternoon, we had a method developed and running because of how much we were able to leverage what we had learned from our previous Orbitrap work onto the Stellar instrument. So I see two areas where there could be a massive benefit to us. One is in reducing that time from beginning a method development project to actually having it up and running. So as metabolomics matures as a field, there are more and more applications that want to move further down that path from discovery to the clinic. The faster we can develop those methods, the better we can serve those researchers, and ultimately, the sooner we can get those things into the clinical world where we can actually be diagnosing patients. Secondly, the scan speed means that any potential existing method can actually be shortened in its duration. And that has a tremendous benefit to the researchers we work with because it takes less time to actually run their samples, and it has a benefit to us as a company because it increases the capacity that we have within our lab, which is simply more and more people we can serve to allow them to leverage the power of metabolomics to answer their questions.